Good morning, students. Today we will discuss about the main questions in sexual reproduction. So that are most likely questions which include 1 mark, 2 marks, 3 marks and 5 marks. Now first we will discuss about 1 mark question in sexual reproduction. So the first question is, differentiate between syncarpous and apocarpous condition. What is syncarpous condition? Now, syncarpous condition means when number of carpels is more than 1, maybe 2, 3 or more than that, when all the carpels are fused. In a simple sentence we can say, the fused carpellary condition, is called syncarpous. Then what is apocarpous condition? Free carpellary condition is called apocarpous condition. So this clearly indicates syncarpous means when the carpels are fused, maybe bicarpellary, tricarpellary or multicarpellary. When the carpels are free, that is called as apocarpous condition. Next question. What is pollen grain? What is pollen grain? Pollen grain is the first cell of the male gametophyte. Which is the first cell of the male gametophyte? Pollen grain, which produces 2 male gametes. The first cell of the male gametophyte, which produce two male gametes, is called pollen grain, or it is also called as microspore. Now in a typical anther, what is the meaning of typical anther? That is dithecous condition. When the anther is two-lobed, that is called as dithecous, that is typical anther. The typical anther always consists of 4 microsporangia, that is called as tetrasporangiate condition. Now, name the different wall layers of the microsporangium. Name the wall layers of the microsporangium. developing anther, or name the wall layers of the developing anther. So which are the protective wall layers of an anther or microsporangium? That are, first is called as epidermis, second is called as endothelium, and middle layer, and the last is called as tapetum. So which are the four wall layers of a developing anther or a microsporangium? That are epidermis, endothelium, middle layer, as well as tapetum. These are the four protective wall layers of the developing anther or microsporangium. Now, the next question is, name the wall layer of the microsporangium which act as nutritive tissue for the development of microspores. Name the wall layer of the microsporangium or anther act as nutritive tissue. So which wall layer of a microsporangium act as a nutritive tissue? That is nothing but tapetum. So tapetum is only the wall layer that provides nourishment for the development of the microspores or a pollen grain. Next one. Name the wall layer of the microsporangium which having a fibrous thickening, that is made up of callous fibrous thickening meant for dehiscence of the anther. Name the wall layer of the anther or microsporangium. Name the wall layer of the anther or a microsporangium with cellulose fibrous wall thickenings meant for anther dehiscence. That is which one? Endothelium. Answer is endothelium. So endothelium usually that having cellulose fibrous thickening in their radial walls. Name the wall layer of the anther where the fibrous thickenings are absent and form a stomium. Again the answer is endothelium. Where the stomium is present in the microsporangial wall. Answer is again which one? Endothelium. Only in the endothelium. cellulose thickenings, but some of the cells of the endothelium which are not provided with this cellulose thickenings, and that involved cells are called as stomial cells. That is mainly meant for dehiscence of the anther at its maturity. That cells are called as stomial cells, or it is called as stomium. Now, name the wall layer of the anther which is persist in the mature anther. Name the wall layer of the anther which is persist in the mature anther. Name the wall layers which persist in the mature anther. So as we know, tapetum usually that way disappear, and which are remaining, that is epidermis and endothelium and middle layers. Now, which layer of the anther cells are multinucleate and resulted by means of endomitosis, that is tapetum. Name the wall layers of the anther. Name the wall layers of the anther with multinucleate condition. Multinucleate condition, which is that again tapetum. Now, so wall layers is over. What is microsporogenesis? As we know, microspores are nothing but pollen grains which are the first cell of the male gametophyte. That is, what is microsporogenesis? It is the process of the formation of the microspores from the microspore mother cells or pollen mother cells by means of meiosis. The process of formation of the microspores from the microspore mother cell. MMC means what? Microspore mother cell by means of meiosis. Only through the reduction on cell division, the diploid MMC, microspore mother cell, that produces microspores or a pollen grain is called as microsporogenesis. Next question. Now, initially, how we can identify or represent the MMC or name the cell which gives rise to microspore mother cell? What is the answer for that? Sporogenous tissue. Name the tissue or cell So usually one of the hypodermal cell in the anther that becomes very conspicuous by its very prominent nucleus as well as dense cytoplasm. That cell is called as which cell? Archaeosporial cell. That archaeosporial cell undergoes periclinal cell division and that results in outer parietal layer and inner sporogenous cells. That sporogenous tissue or cells that act as which cells? Microspore mother cells. Okay. Next question is, what is palynology? So palynology is the branch which deals with the study of pollen grains. Study of pollen grains is called as what? Palynology. Name the two wall layers of a pollen grain are. Directly you can ask the question. What is sporoderm? What is sporoderm? Sporoderm means the protective wall layers of the pollen grain. That is wall layers of the pollen grain are called sporoderm. So the wall layers of a pollen grain are called sporoderm. Name the two protective wall layers of a pollen grain. Which are those? One is called as exine and another one is called as intine. Next question, that is 14th question. Name the two wall layers of the pollen grain. So which are the two wall layers of the pollen grain? One is called as, the outer one is called as exine and inner one is called as intine. Outer exine and inner intine. So the outer wall layer is called as exine and inner one is called as intine. Next one. Name the resisting wall layer of a pollen grain. Which is the resisting wall layer of the pollen grain? Exine. Why that exine is resist to the different physical factors? So it may be sunlight or it may be chemicals called as acid, bases, like that. So because that exine is deposited with the biochemical called as sporopollenin. So which wall layer of a pollen grain resists to the different factors? Name the resisting wall layer of the pollen grain. Which is the resisting wall layer? That is exine. What is sporopollenin? Where it is present? What is sporopollenin? What is sporopollenin? a biochemical which is present in the enzyme that resists to the physical, chemical and biological factors. Sporopolenine, it is the biochemical present in the, it is the biochemical present in the enzyme, resist to the acids, bases and other biochemicals. So why the pollen grains even today we can excavate in the form of fossils because of this biochemical. There is no enzyme on the earth that can destroy this enzyme, that is which one? Sporopolenine. Okay. So which wall layer of the anther that contribute to the formation of this sporopolenine, that is tapetum. Which wall layer of the anther contribute to the formation of sporopolenine. Now look here. Actually, the tapetum is of two types. One is called as the secretory, or we can say glandular, as well as amoeboid. So in case of glandular or secretory tapetal cells, there is a minute bodies called as Ubish bodies. That Ubish bodies of the tapetum that always produces this sporopolenine. Ubish bodies of the tapetum which produces what? Sporopolenine. Now, so somewhere in the enzyme, the sporopolenine is absent. What are that pores called? Yes, germ pores. Next question is, what are germ pores? What are germ pores? What are germ pores? Germ pores are unthickened zones of the enzyme where sporopolenine is absent, and it is the site of penetration of the pollen tube. Germ pores are the unthickened zones of the enzyme where sporopolenine is absent, and it is the site for the penetration, of the pollen tube. So only through this enzyme mainly this pollen tube penetrates. Next one. From which wall layer of the pollen tube emerges? Which wall layer produces pollen tube? In time. Which wall layer of the pollen grain produces pollen tube? Which wall layer produces pollen tube? In time. As we know, majority of the angiosperms, pollen grain shed at 2 cell stage. So, which are those 2 cells? Name the 2 cells of the pollen grain. Name the 2 cells of the 2 cell pollen grain. Which are those 2 cells? One is called as vegetative cell and generative cell. Next one. So, but some angiosperms even that shed in 3 cell stage, that is usually that shows vegetative cell and 2 male gametes produced by the generative cell. That clearly indicates the generative cell of a pollen grain always produces male gametes. Which cell of the pollen grain produces male gametes? That is which one? Generative cell. Generative cell produces what? 2 male gametes. Next one. Name the 3 cells of a 3-celled pollen grain. Name the 3 cells of a 3-celled pollen grain. Name the 3 cells of the 3-celled pollen grain. That is, one is vegetative cell and 2 male gametes. From which cells the male gametes are produced? As we know, it is from the generative cell. Okay. Now, which cell of the pollen grain that is large and always that nourishes the generative cell to produce 2 male gametes? That is nothing but vegetative cell. That vegetative cell with plenty of reserve food material and that only act as nourishing tissue for the nourishing cell for the generative cell to produce 2 male gametes. So that is what about the generative cell. Now, as we know, the pollen grain that act as friend and foe to the mankind. So it has got both advantages as well as disadvantages, and majority of the pollen grains, that pollen grains of the weeds, unwanted plants are called as weeds, that causes different respiratory problems, that is mainly allergy, and also it is responsible for allergic rhinitis, or we can say pollenosis, or even it is called as allergic rhinitis. Now one of the weed, it is called as carrot grass, or it is called as parthenium. So that entered into India as a contaminant along with the imported wheat, isn't it? Now today that is the major cause for allergic rhinitis, or it may be for normal allergy. Okay. Name the seed of a weed that introduced to India as a contaminant along with the imported wheat, that is carrot grass. Name the weed seed introduced to India as a contaminant along with the imported wheat, or simply you can say along with the wheat. Which is that? That is called as carrot grass or parthenium. This entered into our India as a contaminant along with the imported wheat. Next one. Now, as we know that pollen grains are the richest source of what? Nutrients. So that too for the younger generation, this becomes as a fashion to supplement these pollen products. So because of its nutritive value, today majority of the youngsters, they are consuming these pollen products either in the form of a pollen tablets, or it may be in the form of pollen syrup. And so usually this may enhance the athlete performance as well as the horse races. That is one of the major advantage of what? Pollen grain. So in this way we may guess 1 mark questions in the microscope, or it may be in the anther development, or it may be in the cells of the pollen grains. Next is what? The female reproductive part, gynoecium. So in that megasporangium. Now, so megasporangium is also called as what? Ovule. Now, the next important 1 mark question in megasporangium or a ovule. Now what is placenta? Placenta is the what? Tissue present in the ovarian cavity or the locule of the ovary, where actually the ovules are attached. That is called as placenta. So mainly on this placenta, the microsporangium or ovule development takes place. As we know, generally the ovule that having a protective wall layer is called as what? Integument. So generally the ovule having a nutritive tissue called as nucellus. What is nucellus? It is the nourishing tissue of the ovule. Which is the nourishing tissue of the ovule? Nucellus. Now, the ovule is provided with a stalk called as funicle. What is funicle? funicle. The stalk of the ovule, is called funicle. What is funicle? The stalk of the ovule is called as funicle. Now the ovule having what? A nutritive tissue called as nucellus. Now this nucellus is protected by a wall layer called as integuments. What are integuments? What are integuments? Protective layers of the nucellus of the ovule. Protective wall layers of the nucellus of the ovule is called as integument. Now, so the integument that cover all the nucellus part of the ovule, somewhere that leaves a gap, that is called as micropyle. Next question is, what is micropyle? What is micropyle? Micropyle is the zone of the ovule or a nucellus of a ovule where integument is absent. What is micropyle? The zone of the part of the ovule. If it is difficult to write zone, you can write part. The part of the nucellus of a ovule where integument is absent is called as what? Micropyle. Now, the region which is quite opposite to the micropyle is called as what? Chalaza. Now, generally, so the megasporangium development, that too which takes place in this micropyle region of this what